I would like to demonstrate the clinical utility of using our gene dosage maps for chromosome 18. These are helpful because for a typical child, when they're born, you assume everything's normal until something arises, so you know you need to investigate. With the child with a chromosome abnormality, what you end up doing is assuming everything is abnormal and investigating everything to make sure various things are normal. This sort of helps split the difference between those two approaches and helps at least guide a starting place for kids with chromosome 18 abnormalities to understand what things they may be at risk for, what things they're probably less likely to be at risk for. So let me show you a couple of examples here. Here's the first example. This could be a person with a 5.5 megabase deletion from the Q end of the chromosome, and the coordinates are shown here. And so I will show you in the gene dosage map, there's always an ideogram and across the top, and it will have a red box showing some part of the chromosome that's viewed below that. So here's the gene dosage map associated with that region of the chromosome, and the gene dosage map shown here has 3 parts. Here are the UC Santa Cruz genome browser genes. Below that are the gene dosage genes from our custom tracks from our gene dosage maps, and below that are the phenotypes associated with that region. You can see there are 19 genes in this region, and the genes are indicated by little vertical hatch marks that are linked by a horizontal line. So you can see a couple of things here, that the genes are different sizes and they're not distributed evenly. And in this view, they're shown scattered down the view just for readability, because of course you know that they're linear one after the other in the genome. So the genes that are dosage sensitive in our gene dosage map, in this view, we're only showing the ones that have dosage sensitivity, so not the designation of every single gene, but just the dosage sensitive ones. So here's the T-shirt gene, and if you have a deletion that includes this gene, you have a 78% chance of having atretic or stenotic ear canals. The next dosage sensitive gene is MBP, and this is associated with high-frequency sensory neurohearing loss. The next gene is ATP9B, and you'll see it's color-coded yellow here, and it's yellow because it is a risk factor. This gene is deleted more often in people with autism than in people without autism. So it doesn't cause autism, but it may play a role. And so we want to indicate that it has some relevance, but its role is unknown, so it's yellow. The next gene is NFAT-C1, and this gene was significantly correlated with scoliosis. So these are the genes in this region associated with specific phenotypes. So now the phenotype regions. So these are regions where we know there's a gene that causes one of these things, but we don't know which gene, but we have it narrowed down to a region. So these regions are, I'm just showing you with the red arrow here, and the first one is mood disorder. Below that is IgA deficiency, then nystagmus, then congenital heart disease, kidney malformations, vertical talus, dysmyelination of the central nervous system, growth hormone deficiency, and cleft palate, cleft lip. So these are the phenotypes that someone is at risk for if they have a deletion of this region. So what this tells you is at least where to start. And it also tells you those things sometimes associated with 18q- that you maybe don't need to worry so much about. For example, this deletion does not include the TCF4 gene associated with Pitt-Hopkins syndrome. And in fact, here's a person with this deletion of chromosome 18, and here is in fact a little synopsis of their clinical picture. They have an IQ of 120. They are hearing impaired. They have atretic ear canals, or they did have atretic ear canals, and had a foot deformity when they were born. They're growth hormone deficient. They have dysmyelination of the central nervous system. And they also have executive function difficulties, so they need some help and guidance in functioning in daily life, even though they have a high IQ. So that's someone with this terminal deletion of 18q. So let's look at another deletion of 18q. You can see from the red box, it's an interstitial deletion. This is just a little bit larger. The last person's deletion included 19 genes. This one includes 20, so there's a few more genes in the region. And below that, you can see the UCSC genes in this region. And below that, there's the UCSC genes, and below that are the dosage-sensitive genes. So there's only 3 dosage-sensitive genes in this region of 18q. And they are SMAD4 gene, which puts them at risk for juvenile polyposis and hereditary hemorrhagic telangiectasia syndrome. Then the DCC gene, which you can see is a huge gene, and deletion of this may result in congenital mirror movements, but those data are quite limited. So we need to investigate that a little more. And last, which I realize you can't see because the video is in the way, but that's the TCF4 gene, which is associated with much more profound developmental disability. And the clinical picture associated with the deletion of that gene is called Pitt-Hopkins syndrome. And here's a description of an individual with this specific deletion of 18q. At 3 years of age, they had a developmental age of a 6-month-old. At 10 years old, they were not talking, walking, and were not toilet trained. They had a designation of autistic, except, of course, their communication was so limited, so it's mostly around communication, defined around communication reasons. They had the face of associated with Pitt-Hopkins syndrome. But also, importantly, they had normal hearing and ear canals. They were not growth hormone deficient, and they had normal myelination of the central nervous system. These last 3 things are associated not with the deletion of this part of chromosome 18. They're associated with the deletion of the end of chromosome 18. So there's not a reason to investigate them for growth hormone deficiency, for example, unless, of course, they have a growth problem. But you wouldn't expect to find that here. So this really does help define what things associated with 18q- one might be concerned about. In total, there are 263 genes on chromosome 18, and 198 of them we think are variants with regard to gene dosage. So if you have a deletion or duplication of one of those genes, it probably really doesn't have any clinical significance. And that's the vast majority of genes. So those associated with as a risk factor are color-coded yellow in the gene dosage map, and there's 24 of those, so we'll be watching those to try to understand what their role, if any, in causing a phenotype is. But 22 are the ones that we're focused on that we think are gene dosage sensitive, that cause a problem when you have a deletion or duplication of those genes. And then, of course, there's 19, which we have no idea what they do. And so those 19 that are in phenotype regions are ones that are of particular interest to us at this time. So someone's deletion or duplication of chromosome 18, of course, is not going to change over their lifetime, but this interpretation, these data presented here are going to change, and these are updated all the time as we find out more information about people with small deletions, or from our own data, or from animal studies about animals with duplications or deletions of these genes. So these data are going to change, so stay tuned. I hope this helps the management of people with chromosome 18 abnormalities. Thank you so much.